the session on marine viruses that we will have at the next OCB workshop, which will be in early June. So we have in the chat here, Heather, just put the link if you're interested in either attending remotely via Zoom or even in person. And the first webinar is available online, and it was more about why the magnitude and importance of viruses in the environment and their impact on the carbon cycle. Today, we're going to still talk about why we care about viruses, as well as their importance in biogeochemical cycles, but with a more like a virus-host interactions focus. And we have 3 speakers. And I will let Sherry introduce them to you guys.
Great, thanks, Jessica. Okay, so we're going to start with Marcie Marston, who's a professor of biology at Roger Williams University. Marcie studies the ecology, genetics, and evolution of cyanophages. And after each talk, we'll probably have a couple minutes for a question, but then we'll have a larger time for questions after all 3 speakers present. So Marcie, you can take it away if you'd like.
All right. I'd like to thank the OCB webinar series for inviting me to speak today. So for the past 25 years, my students and I have been isolating and characterizing viruses in Narragansett Bay, Rhode Island, which is not far from Hui. We are very interested in the dynamics of viral-host interactions. We know from metagenomic studies that there are lots of viruses in the ocean. However, it's been difficult to sometimes link those viruses with their particular hosts. And so, of course, this is very important if you want to understand the virus impact on host mortality and movement of carbon through the ecosystem. So the model system I work on involves cyanobacteria. The host is Synococcus. These are small unicellular cyanobacteria that are photosynthetic primary producers. They autofluoresce, so you can sort them by flow cytometry or count them. They are culturable, and so it is possible to get individual cyanobacteria clones that you can grow into culture. There are lots of different clades of Synococcus, and they differ in the physiology and the pigments that they have, depending upon the geographical range, right? You get different clades. And here in Rhode Island, in the winter, there are about 10 to the 3rd cells per liter, and the Synococcus that are present are mostly in clades 1 and 4. During the summer, there's a lot more Synococcus present, 10 to the 8th cells per liter, and they're usually found down here in clades CB4 and CB5. Cyanophages are the viruses that infect cyanobacteria. The phages that I work with are all double-stranded DNA tailed bacteriophages, and they also vary in number seasonally, along with their hosts from about 10 to the 5th to 10 to the 8th phages per liter of seawater. This photograph figure is from John Waterbury's lab, who works at Woods Hole, and these are beautiful pictures of the different types of phage. So there are 3 types of tailed phage that can infect Synococcus. And so we'll be talking about these throughout my talk today. There are the T4-like phage that are myoviruses. They're shown in these bottom 2 pictures. And they have relatively large genomes, approximately 180 kb, but they can go up to 250. They often have very broad host ranges, and they carry quite a few genes that they have picked up from their hosts that can influence the metabolism of their host cell, which I'll talk about in a moment. Podoviruses are T7-like phages. They are smaller. They're shown up here in A and B. They have smaller genomes. They tend to carry less of the host-derived metabolic genes, and they often have narrow host ranges. Picture D here is a cyphovirus, and they're very diverse. They have very variable genome sizes, and there are no marker genes or PCR primer sets available to characterize cyphoviruses, so we know a lot less about them than we do myoviruses and podoviruses. So about 20 years ago, led by Debbie and others in the Chisholm lab, when they first started to sequence cyanophage genomes, they found some genes in there that were strikingly similar to the genes of the host. And as we sequenced more and more cyanophage genomes, it became clear that these phage pick up a lot of genes from their host that can help them to modulate or remodel the host metabolism when they infect. And so in this diagram on the top here, it shows the thylakoid membrane with all of the different proteins that are in the electron transport chain and the photosystems. All of the genes here that are in red have been found in cyanophage, picked up from their host, and then can be expressed during infection. And so these host-derived genes can influence photosynthesis, they can influence carbon metabolism, they can influence phosphate acquisition, and also nucleotide synthesis. So those of us who are interested in understanding the dynamics of viral host interactions, one of the first things we look at is how well the phage actually absorb to the cell. So this is often receptors on the tail fibers of the phage and receptors on the cell. It is this stage that often determines host range and sometimes resistance. Then we're really interested in what happens once the virus gets into the cell. And so because these viruses carry all of these metabolic genes that are expressed during infection, they really do remodel the metabolism of the cell, which can have impact on movement of nutrients through the ecosystem. And so as they're infecting, they're influencing photosynthesis, they are changing carbon metabolism, they're making lots of nucleotides, and we call the infected cell a vero cell, and it can have a lot different properties than the uninfected cell. Finally, we're also interested in the number of progeny that are released from one infected cell. And so my lab has been interested in this question of whom infects whom and the dynamics for the past 25 years. And we have been taking two approaches. One is to conduct experimental coevolution experiments in the lab, where we take one host and one host genotype, one viral genotype, and look at them co-evolve over time. And then we're trying to use the information that we gain from these lab experiments to try to understand the dynamics of what is happening out in the ecosystem. And Narragansett Bay is here, Hui is over there, and I'll talk about the research that we're doing to look at infection dynamics of co-occurring Synococcus and cyanophage in Narragansett Bay. So I just want to start really briefly with the coevolution experiments. So for these experiments, these are chemostats, they're continuous culture. We start with one phage, one virus. One phage, one host. The host is susceptible to the virus. When you put in the virus, it increases in number. The cell density goes down of the host, but eventually the host can gain resistance to the virus. It increases in abundance, but then the phage can also evolve to overcome that resistance. What we see over time is that we get coexistence of both the phage and the host. And this is due to a diversification in both the virus and in the host that allows them to coexist. So I'm just going to show two diagrams that are complicated, but the take-home message is that here is the genome of the cell, 7803 cell type. These are genomic islands. Underneath each line is a resistant Synococcus isolate, resistant to one of the phage it was co-evolved with. And each of the black lines is a single nucleotide change. And so the take-home message is that there's not many changes that need to occur in order for a phage to become resistant to a particular virus. All right. And so we have evolved this particular host isolate with all of these different phages. And we see that there's lots of different ways in which the host can become resistant to a particular phage. And it doesn't take many changes. So we also have sequenced all of the viruses that are present after 6 months of co-evolving with their hosts. And here are these viruses. So CAM7 are here. There's two different strains, RM4 here. Now, this is the genome of the virus, right? The ancestral genome. Again, we have mapped the reads for the co-evolved and, in this case, also evolved viruses to the host or to the the ancestral strain. And again, we see that there are lots of different mutations that can occur over 6 months. And it's this diversification that allows both the Synococcus host as well as the cyanophage to coexist, because when you look at the infection network, it's nested. All right. So then we wanted to think about whether we could use this information to understand what's happening out in the bay. And of course, in the bay, it's much more complicated. In our chemostats, we were looking at one genotype of host, starting with one genotype of phage that diversified, right? And here, of course, there are multiple different viruses and multiple different hosts, and there's zooplankton, grazers that can eat the infected cell. And so this system is much more complicated. We thought about this in terms of this paper that was written by John Waterbury in 1993. And he did this work at Hui, and it's a wonderful paper. And basically, he went out and he isolated cyanophage and also Synococcus from the same location at Woods Hole Harbor, and he crossed them, and he came up with this infection matrix that showed which phage could infect which host. And from that, he determined or wrote that the resistance to co-occurring Synococcus is common, right? And it allowed them to coexist. This was done in 1993 before sequencing was really possible. And he looked at just a few different strains of Synococcus and strains of cyanophage. And so working along with Nathan Algren at Clark University, we decided 30 years later to try to repeat the study, but using all of the omics tools that we now have. And so this is Nathan and our students. This is our dock where we've been collecting seawater, and Nathan has collected seawater, taken it back, and has isolated Synococcus strains from the bay. At the same time, we took the same seawater and we isolated strains of cyanophage. We're then doing host range assays where we take one Narragansett Synococcus strain, and we look in this picture at 10 different viruses. And so here in red would be virus A that can infect the first host and cannot infect the other two hosts. Right? Here's like virus B that cannot infect the first host. It can infect this one. It lyses it, right? The cells are no longer green. And then it cannot infect this Synococcus 3, right? And so what we're gaining here is a host range of each of the different viruses on each of these different types of cells. In addition to this, we are also fully sequencing all the genomes of all of the viruses that we've isolated, all of the hosts. We have done amplicon sequencing of both hosts and phages on a weekly time series for 2.5 years, and we have metagenomic and metatranscriptomic data to go along with this. And so hopefully by putting all of these together, we'll have a better idea of who infects whom and the consequences of those infections in Narragansett Bay. All right, so next I'm going to show you what the infection network looks like after we did all these host range assays by taking individual phages and seeing whether they can infect particular Narragansett Bay strains. So in this matrix here on the left, we have the Synococcus. And so the ones that are in orange and purple are the Narragansett Bay Synococcus that Nathan has isolated. These are done in the summer, and so they typically fall in CB4, CB5. We're also testing against common strains of Synococcus in other clades, as well as Prochorococcus. And you can see that we've isolated a number of Synococcus strains that have identical ITS sequences. On the top here, we have the cyanophage. These are all myoviruses or T4-like viruses. They were classified at first based on DNA polymerase gene sequences. And again, we'll see that there will be a number of viruses that have identical DNA polymerase gene sequences. But there's also diversity, right? In one water sample, we can pick up easily 20 or 30 different types of T4-like cyanophages that can infect these. So when we look at this matrix, the black indicates that this virus could infect that cell. Blue indicates which host the virus was isolated on. White indicates that there was no infection. And so indeed, just as John Waterbury saw that there were some Synococcus that were resistant to co-occurring phage, we also see that. But we do see that there's quite a bit of infection. What we're interested in are these cases where we have viruses that have identical DNA polymerase gene sequences and cells that have identical ITS sequences and yet have different infection, right? And so this is similar to what we have seen in chemostats. And so we're really interested in looking at these cases where we have very closely related viruses and that infect very closely related cells, and yet we see some are resistant and some are susceptible. And so to investigate this, we're doing infection experiments in the lab where we take the cells that were isolated from Narragansett Bay, we take the co-occurring virus that was isolated from the same water sample, we put them together into small test tubes. We do this in replicates of 4. And at various time points, then we take out cells, or take out an aliquot, we filter out the cells. So we are just left with phage that are complete virions, right? Or loose DNA. And what we're filtering out is then any phage that's attached to or stuck to cells. And so we are using a qPCR cybergreen assay to look at the dynamics of infection. And I'm just going to walk you through what one of these might look like. And so in this case, we have, we normalize to the amount of extracellular DNA relative to time 0 here. And again, this is determined by qPCR assay looking at the DNA polymerase gene of these viruses. At first in infection, first during infection, the number of viruses decreases, and that decrease occurs because the phage is attaching and entering the cell and starting to go through replication. We are filtering these all out, and so we see a decrease in the amount of phage, extracellular phage in the media. When the phage start to lyse, then we see an increase, right? And then this is sort of the second cycle. So this is over 48 hours. This is a semi-log graph. So here, I just want to show you just sort of one example. We've done many different pairs, but this is one example. These are 2 cyanophage that were isolated from the same water sample. We sequenced their genome. This is a MAV alignment of those 2 genome sequences. This is each a different gene. And above it, if there is a blank, it suggests that one virus has a gene that the others doesn't. All of these little yellow lines are SNPs present in these 2 genomes. But overall, these 2 genomes are very similar to one another, yet there are some differences. And here we see that Synococcus 1 can infect the cell type, or that cyanophage 7 can infect Synococcus 1, but cyanophage 14 cannot. And so when we did the qPCR and we looked to see what happens, indeed, this is what the qPCR shows, that in this susceptible reaction, the virus attaches, it replicates over 48 hours. In the resistant reaction, interestingly enough, the virus seems to attach, but there's no replication. So then we looked at these 2, in which interactions with Synococcus isolate 2, where they both look susceptible, but it's not, complete lysis as we see with the Synococcus 1 cell type. And so when we look at those, again, we see that, yes, they both can infect. There's attachment, and then there's replication over 48 hours, but the dynamics are different. And so actually, the cyanophage 14 has a stronger attachment, right? And a longer latent period before it starts to replicate, and less progeny produced, at least in the first 24 hours, than the other CP7 on Synococcus 2. And so we're trying to understand these dynamics of how each of these different phages interacts with these different hosts. They're all co-occurring. There are suboptimal hosts. There are hosts that it may replicate better. It's bumping into all of them, right, in the environment, and trying to understand how that influences both the long-term stability and overall mortality of these viruses. I just say that when you're thinking about trying to study the dynamics of these interactions, certainly there's something to do with genetics, right? So someplace in here, the differences between these two viral isolates, allows one to infect Synococcus 1 and one to not be able to infect Synococcus 1. But environment can also play a big role. And I haven't talked about that, but if you change the light levels, if you change the temperature, you can change both the latent period, the attachment, and how much virus is actually produced. And my guess is that there's also genotype by environmental interactions that will, again, influence the outcome of these interactions between the Synococcus and cyanophage. And so, I mean, this is an ongoing question about who infects whom. I think the dynamics of this viral host interactions is super interesting and complicated. And again, I think that, we do see long-term coexistence of these viruses and hosts, likely because there's this microdiversity present both in chemostats and in the natural environment that, allows some stability over the long term. And so I'd just like to thank my funding sources and all my collaborators, and I will stop here.
Thanks, Marcie. That was amazing. That's a lot of data to show. So I guess we have maybe 1 or 2 minutes if anyone has a burning question they'd like to ask. Okay, I have a list in my paper, but I'm gonna hold them till the end. Oh, Debbie, okay, go ahead.
Yeah, I can ask a question. Always got questions. Do you have any idea, that place that's different? What's different? What type of gene is it? Especially if both of the if the phage attaches to both phages attach.
Right. So those are one of the genes that we don't know, right? They're hypothetical, right? They're hypothetical or that we don't know the function of. But yeah, we're waiting for your students to figure out actually why they're important in host interactions. Yeah.
Yeah, you bring up a lot of the challenges we face in marine virology, and especially linking viruses to ecosystem scale processes, which is one, we have all of these unknown genes, but then also the infection dynamics change pretty rapidly with evolution, but then also with environmental parameters. So trying to get to a place where we can predict how these interactions may change with climate change is certainly a big task. Okay, so I'm gonna turn it over to Debbie Lindell, who's our next speaker. She's a professor in the Department of Biology at the Technion Israel Institute of Technology, and she studies the role of cyanophages in ocean ecological processes.
Okay, can you see my presentation mode? Terrific. Hi, everyone. So I'm going to continue our discussion on host virus interactions in marine cyanobacteria, but I'm going to present somewhat different data looking at the distribution patterns of cyanophages and the pattern of their infection across gradients in the North Pacific Ocean. And I'm going to particularly focus on a hotspot of cyanophage infection that we found in the North Pacific Ocean. And so as Marcie has already introduced to you, there are two major types of picocyanobacteria that we talk about. These are Synococcus and Prochlorococcus. Together, they contribute significantly to primary production, but they're different organisms. They have different pigment contents, and they have perhaps most importantly, they have different distribution patterns in the oceans. When we think about the ecological role of viruses, we think about three major things. Think about how they regulate host populations. Because they infect and kill their cells, they can affect the abundance, and therefore we're talking about primary producers, the amount of primary production they do. And because they can be specific, they can impact the community structure as well. They're also a driving force for host diversity and evolution through the selection for cells that are resistant, similar to what Marcie's just told you about. And they also are vectors for horizontal gene transfer, moving genes from one organism to another. And perhaps most relevant for this audience is their role in carbon cycling. And we think about them as important for recycling organic matter in the photic zone, particularly when they're infecting autotrophs and transferring that material to heterotrophs. And Kim will probably also be speaking to you a little bit about their role in carbon export. So the marine cyanophages, as Marcie told you, are double-stranded DNA phages. Most of the ones we all work with are lytic. There's two major families, the T7-like potoviruses and the T4-like myoviruses. There are other types, including TIM5-like myoviruses, and I will speak about these in today's talk. Also, as Marcie has already told you, they differ in morphology, they differ in their genome content, they differ in their infection physiology, they differ in their host range, and in fact, the breadth of their host range, and they're highly diverse. So you can see here a tree of T7-like cyanophages based on the DNA polymerase gene, and you can see that there's two major clades, clade A and clade B. They differ in the types of special genes they carry from their hosts, thyrodoxin for clade A, photosynthesis gene, PSBA in clade B. And they also alter in their infection properties. Clade B has a longer infection cycle and lower burst size than clade A phages. And so we think, and this is just an example for the T7-likes, there's going to be examples like that for the T4-likes, but these differences between different clades of T7-likes and between T7 and T4-likes are likely to impact their distribution pattern and the way they infect cyanobacteria in the environment. This is what we want to address. So our research questions that we want to deal with today are: how do cyanophages respond to changing environmental conditions and microbial distribution patterns? And how do cyanophages impact cyanobacterial communities? And our approach is to quantify both cyanophages themselves and their infection patterns across transects in the North Pacific Ocean, going from Hawaii northwards. And we'll also look at the cyanophage diversity as well as cyanobacterial diversity across those transects. And we're going to focus really on three major regions: the subtropical gyre, close to Hawaii, the subpolar gyre, and the transition zone between them. Okay. So there's changes occur across these transects, and we'll look at first at the left, the environmental conditions. We can see that temperature declines along the transect. On the x-axis, we have the latitude. So temperature declines, as does salinity. Nutrient levels increase as we go north on each of these transects. And also, we have particulate carbon, which is an indicator of trophic status, also increases as we go northward. What I'd like you to notice, though, is that these increases differ at different places for different years. And therefore, we're plotting here what's called the chlorophyll front, 0.2 milligram per meter cubed concentration of chlorophyll, which indicates when we go from a less productive to a more productive body of water for each of these three cruises. I should have mentioned that each cruise is a different color here. And so we can see when each of these transitions from low productivity to higher productivity. And now if we have a look at phytoplankton abundance on the right, we see that Prochlorococcus is highly abundant in the subtropical waters, that Synechococcus is more abundant in the region of this chlorophyll front, and that the eukaryotic phytoplankton become more abundant as we go further north. What I'd like you to notice already is that for two of the cruises, Prochlorococcus declines north of the chlorophyll front, whereas in one of the cruises in 2017, June 2017, Prochlorococcus declines significantly south of this chlorophyll front. Okay, so how do we want to measure the distribution? To look at the distribution pattern, we need to be able to measure the abundance of the cyanophages as well as their infection. And this is far from trivial, but I'm not going to go into the ins and outs of it right now, but just to say that we've developed a method based on a single molecule PCR assay in solid phase that allows us to quantify diverse groups of cyanophages at at a functional group level or a genus level or family level. So we use a target gene, like the DNA polymerase gene that Marcie talked about, to amplify these cyanophages, and then we use a probe to detect the amplified DNA from the viral fraction, and then we can count the number of viruses of T7-like or T4-like in our samples. Perhaps more importantly, we can use this method in a similar fashion to determine and quantify infection. To look at infection specifically of Prochlorococcus and Synechococcus, though, we first sort the cells using a flow cytometer based on their pigment content. So we sort Prochlorococcus and Synechococcus into separate tubes and then embed them into a gel solid phase PCR separately. And then we do the amplification for viral DNA so that we're detecting viral DNA inside cells. And we do this at a high number of cells, so we've got we can go down to very low levels and still have statistical robustness. And so when we use this method to assess the distribution of cyanophage groups, we see that we have T4-like cyanophages that are abundant in the subtropical gyre, that we have the T7-likes that dominate in the transition zone between the two gyres, and that the TIM5-like group becomes more abundant as we go north. But what I'd like you to notice, besides this consistent community composition at the family level of the cyanophages, is that in this transition zone between the subtropical and subpolar gyres, we have an increase of both T4 and T7-like cyanophages, but this increase in T7-like cyanophages is higher. And we're calling this region, we've marked in grey, as a cyanophage hotspot. And you can see that based on latitude, the position of this hotspot is different in each of the cruises due to differences in environmental conditions and annual differences as well. And when we plot these data with temperature, we see that the hotspot of cyanophage abundances occurs at a consistent temperature between 13 and 18 degrees Celsius. We're not saying here, though, that temperature is driving this, but rather that there's a correlation between the hotspot and temperature. Okay, now temperature is known to be an important parameter in limiting cells in general and in Prochlorococcus in particular. This is work by Eric Zinzer and showing the temperature range of Prochlorococcus and their abundance in the field in these open circles and in their growth rate of isolates in the line and closed circles. And we can see that below about 11 or 12 degrees, Prochlorococcus doesn't grow and is rare, if abundant, if present at all, in water samples. So when we have a look at our data from two of the cruises, we see that the decline in Prochlorococcus matches the decline, the expected decline, based on their temperature optimal or temperature limitation to growth. But in the third year of 2017, June 2017, the decline in Prochlorococcus happens at considerably higher temperatures than would be expected, and certainly at temperatures that they can grow well at, indicating that something else is going on in that particular year. Okay, so one thing I wanted to mention also is that we can see that the decline in Prochlorococcus in 2017 here in the orange, it correlates with an increase in cyanophage in the hotspot, and that in 2017 we had 500,000 more cyanophages per milliliter of seawater that could be causing this. So that's a correlation, though, and if we want to have a look at measurement of infection, we use our method to look at viral DNA inside infected cells. And when we do that for each of the 3 years, we see that in the hotspot we have a peak of infection of both Prochlorococcus and Synechococcus, but that in the June 2017 year, we have a considerably higher increase than in the other 2 years, reaching up to 10% of the cells of Prochlorococcus and Synechococcus being infected. Those are instantaneous measures of infection, how many cells we actually measured at a particular time. But then if we want to average that over a day to see how much daily mortality there is likely to be due to viral infection, we take into consideration that a number of infection cycles can occur during a day, and we reach 20 to 30% of Prochlorococcus and Synechococcus cells could be being killed by viral infection in June 2017. If we then calculate this relative to the division, the time it takes for a cell to divide, because Prochlorococcus takes considerably longer to divide at those temperatures than Synechococcus, then anything up to from 20 to 50% of Prochlorococcus could be being killed prior to cell division. And so Synechococcus might be surviving better under those conditions, despite the fact that they're highly infected, because less would be killed off prior to division because they grow more quickly. Okay, so what are the potential biogeochemical implications of this high infection in June 2017? So what we all assume is that viral lysis causes the release of the organic matter that's inside cells that can then be taken up by heterotrophic bacteria and used for their growth. And in fact, we see that bacteria their abundance increases in the transition zone. I'm not saying it's just because of viral lysis. They can also, the increase in nutrients, inorganic nutrients, etc., can be driving this. But when we take the amount of lysis, if we estimate the amount of carbon in those lysates, we see that in subtropical waters of 2017, viral lysis would only be able to provide for about 4.5% of the demand needed by bacteria, which has been determined by leucine incorporation experiments. Whereas in the hotspot, we see that the viral lysate produced could contribute up to 30% of the bacterial demand. So there could be considerable consequences to such a high lysis event. And what we have here is enhanced recycling and transfer of organic matter fixed by autotrophs moving over to heterotrophs in 2017, or at least that's what we think is going on. Okay, so now let's transition to the next part of the talk. We want to look at the diversity of both the cyanobacteria and the cyanophages. And to do this, we're looking at metagenomics, first of the viral fraction, what we call the viromes, in the 0.2 micron fraction. And then we're also looking at viruses in the cellular fraction, in the 0.2 to 3 micron fraction, which are viruses that are infecting cells. And we're also looking at cellular diversity. And we've also done metatranscriptomics, but metatranscriptomics and metagenomics have shown pretty much the same results. So I'm just showing you the metagenomic data. And we make use, once again, of marker genes to have a look at the diversity of cyanobacteria and of the two major groups of cyanophages, T4-like and T7-like. And we're going to be looking at a clade level for both the cyanobacteria and the cyanophages, large groups, and also genomic or genotype diversity, phylotype diversity that's at the leaves of these trees as well. Okay, so these are bar graphs. I'm just going to, these are relative abundance of Prochlorococcus ecotypes at different stations along our transect, and I've broken this down into three groups: the subtropical area, the hotspot area, and the northern transition zone. And what we can see is that we have consistent shifts in Prochlorococcus composition on each of the 3 years. Highlight 2 types for those of you who are familiar with them, are abundant in the subtropics. Highlight 1 types are abundant in the more colder waters. When we then have a look at the phylotype level, the higher resolution genotype level of these types, we see that there's a large degree of diversity in the subtropics, perhaps less diversity in the hotspot, and that we have 2 subtypes of highlight 1 types. We have 1 type here, the light blue, and the dark blue type. And these lines here indicate the abundance of Prochlorococcus in the upper graphs and percent infection in the lower graphs. And what I want you to notice is that we've got a decline in Prochlorococcus, and you can only understand sorry, here we have a transition from the highlight 1A type to highlight 1B type that you can follow at the phylotype level, and you don't see that when you look at the ecotype level. And this transition is accompanied with the increase in infection, suggesting perhaps that cyanophages are infecting these this particular type and killing them off, and causing a difference at the phylotype level in the distribution of the Prochlorococcus types. We have a similar type of dynamic of Synechococcus, consistent distribution over the years, and I'm, in the interest of time, will not belabor that. Now, if we look at what's going on with the cyanophages, first of all, we're looking at what's in the free viral fraction in the viromes versus what's infecting cells in the metagenomes. And what we can see is that we have different T4 subclades and different phylotypes that are infecting cells that are in the free phage fraction. And that's probably most clear from the T4s. Here we have this purple phylotype that is quite rare in the reservoir, in the viral fraction, but very abundant in infected cells. Okay, what we also see is that there's greater phylotype diversity in the free virus fraction than in the infected fraction, suggesting that there's a large reservoir of viruses that are not infecting, and opportunistic viruses that are getting into cells and infecting and causing a decline. And what we also found was that we have different phylotypes, particularly of the T4-like cyanophages, that are dominating in each of the years. So what I showed you previously was in the distribution was of gradients 2, but if we look in gradients 1, we have a different type relative to gradients 2, relative to gradients 3, suggesting that there's some sort of stochasticity at the phylotype level that doesn't exist at the higher, at the subclade or clade level. Okay, now if we look at an overview of these spatial differences in phylotype diversity, we see that Prochlorococcus diversity is high in the subtropics, and it declines as we go north, and we see a similar pattern for Synechococcus. When we have a look at the cyanophage diversity, we see a bit of a different picture. For the T4-likes, we have high diversity throughout the transect, but in the 2017 cruise here in the red dots, we see that diversity dropped, and that's the year where we had the high degree of infection by both T4 and T7-like cyanophages. And different again, we have a very different picture for the T7-like cyanophages. Their diversity is lower in the subtropical waters, and it increases as we go north. In the interest of time, I'll skip that one and just sum up to say that we found that there are distinct cyanophage community compositions in different regions at the genus level, but not the phylotype level, that we have a hotspot of cyanophage abundance and activity in the transition zone between the subtropical and subpolar gyres, that we have a virus reservoir that appears to be quite different to the infecting viruses, certainly in regions of high viral infection, that cyanophages can have a large impact on picocyanobacterial diversity, abundance, and biogeochemistry, and that there are downstream impacts of these large infection events on biogeochemical cycles and heterotrophic community. And I just want to finish up by thanking the people that did the work, primarily Michael Carlson and Kira Kondrateva, as well as our collaborators from the University of Washington and the University of Hawaii. And thank you for listening.
Thank you, Debbie. That was a tremendous amount of data. It was really exciting to see. I think maybe in the interest of time, we'll hold questions until the end, and we'll move on to Kim. And so we're going to switch gears a little bit and talk about viruses infecting eukaryotic phytoplankton, moving away from the cyanobacteria. And so I'd like to introduce Kim Thamatrakoln, who's an associate professor in the Department of Marine and Coastal Sciences at Rutgers University. And Kim has been doing work with viruses that infect both diatoms, which I think she's going to talk about here today, but also some work with coccolithophores. And she's particularly interested in phytoplankton molecular ecophysiology. And so I'll leave it at that.
Great. Can you see my slideshow?
Kim, I think you're in presenter. You want to,
I did this before. I did the exact same thing I did before. Let's see. Yeah, I said, oh, does this work?
it worked before, but it's not.
Yeah. Still see the presenter mode.
Yeah, we see the,
Yeah.
Can you change your screen? So you've got two screens, you might want to switch the screens.
Yep.
You can also click at the top on presenter settings and just mirror your
Right.
presentation.
Is that better?
Yep.
Yes, you got it.
Okay. Great. Thank you so much. I really appreciate the opportunity to talk to you. I'm going to take, like we said, we're going to switch gears. We're going to talk a little bit about eukaryotic host virus systems, focused mainly on diatoms. And I'm going to take a little bit of a different approach. I'm not going to show as much detailed data, but rather I know that one of the goals of the summer OCB workshop and the session on viruses is to try to understand or just, start to talk about ways that viruses can be incorporated into ecosystem models. And so the goal of this, my talk, is really just to highlight some of the things that we think about when we're considering how to incorporate viruses and their interactions with hosts into these models. And so this is this is a global ecosystems model from the MIT Darwin project. And these kinds of models have been really valuable in helping us understand the distribution of phytoplankton groups within the global ocean. And one of the goals that I think a lot of us in the community are interested in is how do we incorporate the role of viruses? So this is a somewhat famous picture by Jed Furman of a drop of seawater that's been stained with the DNA fluorescent dye cyber. And what you're seeing here is you can see a diatom cell here off to the right. These larger dots are bacteria, and then all these smaller dots in the background are viruses. And so viruses are the most abundant entity in the ocean. And so one of the ideas is one of the things that we're interested in is to understand how viruses may interact with their hosts and drive ecosystem processes. And so what do we need to know in order to incorporate viruses into an ecosystem model? And this is a very sort of simple, a somewhat simplified view of some first order parameters that might be needed in order to start to incorporate viruses into these models. And you've heard about these in some of the first two talks, but knowing viral abundance, images like I just showed you in the previous slide on with the cyber green only really show you the total abundance of viruses. They don't really get at the total infectious particles. And so knowing how many viruses are actually capable of infecting a host is important. We need to know the spatiotemporal distribution of viruses and their hosts, so where they are and when. Infection rate, which includes the encounter rate, so how frequently a virus will encounter a host, the rate of mortality, how fast a virus can actually kill its host, burst size, the number of viruses produced by a host, and then the virus decay rate, so how quickly viruses actually decay. And so this is a somewhat simplified view, and as many of my modeler colleagues have told me, biologists and biology tend to complicate this image, this picture. And so when I think about host-virus interactions, I think of all the different aspects of the environment that could impact these different interactions. And we've heard a lot about them in the cyanophage system that we just heard from Marcie and Debbie, and how the environment can really dictate how these interactions manifest. And so we think about sort of the physicochemical conditions, so the nutrient background, the physical conditions, so the microscale turbulence that dictates the encounter rates. But then we also think about sort of the diversity and the abundance of these host and viruses, virus communities, impacts of host physiology. And then there's a whole virus life history, temperate viruses versus lytic viruses. And I would argue that, some of these factors probably the details of these aren't really necessary to incorporate viruses and their action into ecosystem models. But I would argue that we need to understand some level of the biology, because I think we need to know, in order to understand where, how viruses are impacting carbon flow in the ocean, we need to know what the consequences of those interactions are, whether viruses will shunt organic material away from higher trophic levels and back into the microbial loop, or whether they'll actually stimulate host processes that could facilitate particle aggregation and possibly sinking and carbon export through the viral shuttle. And I think knowing where the carbon is going and knowing sort of, what viruses are doing in terms of that carbon flow is really critical if we want to be able to understand what the role of viruses are on ecosystem level. So I'm going to use the backdrop of diatoms for this talk. Diatoms, as many of you know, are unique phytoplankton because they make a silicified cell wall called a frustral, and this silica-based cell wall acts as a ballast mineral, and so diatoms just naturally sink because the silica is heavier than the density of seawater. And I've always been very interested in diatoms and their role in silicon and carbon cycling, and I started thinking about, many years ago, about the role that viruses play in interfacing in interacting with diatoms and how they might impact silica production and silica regeneration. But for a long time, diatoms are actually thought to be immune to viral infection due to the physical protection afforded by their silica cell wall. But the first diatom virus was actually discovered back in 1999, and it turns out that they're just they've been able to evade sort of our methods of detection because they are among the smallest viruses on the planet. And so this is a size scale image of different viruses. Most of them are algal viruses, with the exception of this Pandora virus shown here on the right, which is the largest virus known on the planet. This infects a freshwater amoeba. And then for context, this is the SARS-CoV-2 virus. So this is the virus that has been the cause of the COVID-19 pandemic. The rest of these are algal viruses. And so we see that the diatom viruses are all the way here on the left. Their virions are about 20 to 40 nanometers in diameter. But what also makes them unique is that among these viruses here, all the algal viruses shown here have genomes comprised of double-stranded DNA. Whereas diatom viruses have genomes that are comprised of single-stranded RNA, similar to the SARS-CoV-2 virus, but also viruses that have genomes comprised of single-stranded DNA. And so when you look at images like this, using fluorescent dyes that are specific for double-stranded DNA, they completely, they ignore the contribution of diatom viruses. This is also epifluorescence microscopy, so it's difficult to get resolution at the size scale of these viruses, which I said was about 20 to 40 nanometers. So if we just do a back of the envelope calculation, we think about the average viral abundance in the ocean from images like this, we can estimate it to be about 10 to the 7 per mil. And if you take a diatom bloom, a typical bloom may get to about 10 to the 4 cells per mil. And if you consider the number of viruses produced from a cell, this is an average number that we know from model host virus systems that we have in culture, you can get about 10 to the 4 viruses produced per cell. So if only 10% of a diatom bloom is infected, that would produce 10 to the 7 viruses per mil, which is rivaling the concentration of the viruses that we can see through this method. And so just getting at sort of first order how many viruses are in the ocean, I would argue that we don't really know. These estimates I think are severely underestimated. So one of the things that we've been interested in is understanding the impacts of nutrients on silica production. So the silica is that ballast, and so we have an interest in understanding carbon and silica cycling in the ocean. And we started looking at this. We started this work by looking at 2 different contrasting nutrient regimes that have varying impacts on silica production. So we know that diatoms experience widespread silicon and iron limitation throughout the world's ocean. And we know that from culture work that diatoms under silicon limitation actually decrease silica production because they lack the substrate needed to make that silicified cell wall. But under iron limitation, diatoms, at least in some species, can actually increase silica production. And so we wanted to understand how viral infection may interact on under these different nutrient scenarios. So this was work that was done by my former postdoc, Hannah Kranzler, who is now a professor or senior scientist at Bar-Ilan University. And we started doing this work in a host virus model system that we have using the diatom Catastris tenuissimus, for which we have two viruses that infect it. We have a single-stranded DNA virus and a single-stranded RNA virus. And one of the things Hannah did was she just grew Catastris under different nutrient limitation scenarios. And this is just a time course of infection showing host abundance under replete conditions, silicon limited or iron limited conditions. And what we saw was that under silicon limitation, which is shown here in green, the infection was faster. The cells were more susceptible to infection, in contrast to under iron limitation, where the cells were less susceptible to infection. The latent period was much longer and the time to lysis was much longer. This also has an impact on the burst size, so the number of viruses that were produced per cell, where the cells that were infected under replete or SI limitation produced roughly around the same number of viruses, but there is much fewer viruses produced under iron limitation. So if you go back to that first slide that I showed about just knowing how many viruses are produced, what the time to mortality is, we see that there's this dynamic shift depending on the nutrient limitation scenario. And this is likely due to an impact from the host ecophysiology. And so this leads us to sort of this hypothesis that there are nutrient-driven biogeochemical consequences of viral infection mortality in diatoms, whereby diatoms that are infected under silicon limitation might be more prone to shunt material back into the microbial loop because you're causing a faster infection, whereas that infection under iron limitation may cause diatoms to actually sink and be exported into the mesopelagic. So we've recently started looking at quantifying processes that facilitate, that would tip the balance towards the viral shunt or the viral shuttle. And this is a very simplified view. There's a lot of other factors that probably contribute to both of these sides, to both of these aspects. But things that we've been looking at, particularly in my group, is looking at sort of aspects that impact the viral shuttle. So this would be an increase in ballast production. And so we see that infected cells have a higher cellular silicon to carbon ratio. And this is seen during the early stages of infection, pre-lysis. And so we see here a time course of just host abundance of infected cells shown here in orange, and then the elemental stoichiometry of silicon to carbon in those infected cells is higher than that which we find in uninfected cells. And so this would represent sort of an increase in that ballast mineral that could facilitate export. We've also seen that viral infection can induce spore formation in diatom species that are capable of forming spores. This is work that we did in collaboration with Marina Montresor in Catastris socialis, where we see that when we added viruses to a culture, we saw that up to almost half of the culture transitioned from the vegetative cell to this resting spore stage. And resting spores in diatoms are very, very heavily silicified, so they sink rapidly out of the mixed layer, which is another mechanism that could facilitate carbon export. We've also seen that viral infection can induce aggregation and large particle formation. So this is work that was done by colleagues, Yamada et al. In Catastris tenuissimus, where they see that there's a larger particle size diameter that's produced when cultures are infected with viruses. We've seen similar results in our own systems with Catastris socialis, and so this is a large particle. This is a particle that's been stained with Alcium blue. So this stains transparent exopolymers, which are sticky polysaccharides, and you can hopefully see all the cells that are sort of included within this large particle. And these large particles have a higher propensity to sink, which would be another mechanism by which viruses could stimulate the carbon export. In terms of looking at aspects that stimulate the viral shunt, we've been focused mostly on the impact of viral infection on the microbial loop. And so bacteria are responsible, as we saw in Debbie's previous talk, looking at the ability of viral lysates to support secondary production by bacteria. And one of the things that we've recently seen is we started looking at the dissolved organic matter that's produced during viral infection by different viruses. And what we see is actually a very different impact of that infection on microbial activity. So when we take our host model system and we infected it with a single-stranded DNA virus, we see that the DOM that's produced during that infection is able to directly support secondary production and microbial growth. In contrast, when we infected with a single-stranded RNA virus, that DOM was not actually bioavailable for secondary production. Bacteria couldn't actually grow off of it, and actually they needed to actually induce the activity of proteases in order to hydrolyze that material to be able to use it for eventually for growth. And so we see this differential impact whereby these two different viruses are both lytic viruses and they're causing a lytic burst and the production of DOM. But in one case, that DOM is available for secondary production. And in another case, that DOM is not. I mean, so it adds this layer of complexity to the role that viruses play in recycling material. We think that this, and this is supported by recent work that we just published. This is work that we did in collaboration with Bethany Edwards and Ben Van Noy, looking at the dissolved lipidome of viral lysates that are produced during infection. So what you're looking at is a heat map of just a subset of dissolved lipids that we were interested in looking at that are produced during DNA virus infection shown on the left and RNA virus infection shown on the right. And the point to just highlight is to just highlight that there's a different signature of dissolved lipids that are produced during these different infections. And so even when we think about just viral infection causing a lytic burst and an increase in DOM, knowing whether that DOM is actually available for secondary production, I think is important for us to be able to include these include these entities into ecosystem models. And then the last two slides. So I wanted to just mention two projects that we have ongoing in the lab right now. So this is a project that is funded by one of NSF's 10 big ideas, which is a growing convergent research project to look at the impact of marine viruses, minerals, and microscale physics on phytoplankton carbon sequestration. And so we're looking at specifically diatoms because of their silicified cell wall, but as well as copolithophores with their calcified cell wall. This is a project that's led by Kay Beidel here at Rutgers, and it includes 10 other PIs from 7 different institutions that are all looking at sort of the interface of how physics increases the encounter rates between viruses and their hosts, how that manifests in terms of biological interactions between host viruses and host engrazers, and what that does for the mineral ballast of each of these respective organisms, the silica and the calcium carbonate. And we actually have our first of 2 cruises that's going out next week. So I leave on Wednesday for a month-long cruise in the California Current, where we're actually going to be bringing a lot of our methods together to try to look at the impact of viral infection on particle aggregation and sinking. And then the last thing I wanted to talk about was just this project that we have in collaboration with Juan Botticello, who's a theoretical ecological modeler here at Rutgers. And Juan is really interested in understanding viral plasticity. So we've been talking a lot about sort of the role of host ecophysiology on viral infection, but there's also a plasticity that's associated with the viruses themselves. And so the ability of viruses to actually acclimate to host physiology and modify these different parameters of the infection cycle, whether that be the latent period or the burst size. And we saw that in some examples, again, shown previously in the previous talks, where we see differences in the latent period and the burst size under different conditions. And this has implications for host abundance. I'm not going to go into this particular work, but we've been working with Juan over the past few years to understand what the role of viral plasticity is among different algal viruses. And so that's a very sort of like 30,000 foot view of the things that we think about when we're trying to think about the role that viruses play in host virus interactions. And so trying to understand some of these aspects, I think is important if we're hoping to try to incorporate these aspects into ecosystem models. And that is all I have. Sharing that.
Thank you, Kim.
Yeah.
That was fantastic. I really appreciate that. Okay, I'm going to open the floor up to questions for Kim, Marcie, or Debbie.
I can ask one. David, tell me here. I thought I'd go back to the first talk. I may have missed this, Marcie, but I was curious. In your chemostat data, you get those kind of intermittent periods. There's the initial drop in the hosts, right, and then you kind of have this coevolution happening. I was just wondering if you had any sort of speculation about. This probably could also be asked to Debbie as well, but what might be driving this kind of intermittent nature of things? Do you have any sort of, gut feelings about what might kind of trigger greater susceptibility to viral infection?
So when we first put in the phage with a susceptible host, why it drops and then it comes back? Is that you're asking that very initial stage or later on?
I guess I'm sort of using the data as inspiration because it seems like there might be some intermittence in this, just generally speaking, right? And it just seems like kind of a mystery to me. And I'm just curious if you have any thoughts about if there's something in the environment, what might be. I realize that we probably can't answer that definitively, but just more generally speaking, what might be driving some of this of things.
Yeah, I mean, that's a good question. I think, initially they're mostly susceptible. The cells are mostly susceptible. We do see a drop. I think that when we start, those cells have been, start off as a single genotype, but they've been growing in that culture for 2 or 3 weeks to become stable. So I think even in that short amount of time that there are differences, right, point mutations that have occurred. And after, I don't know if this answers your question, but after the virus infects, most of them are susceptible, but there are still some that are capable of resistance, right? And then those are slowly growing back. We do see that there are, as we go through time, that it wavers, right? It goes down and up, and we do see that, we can sort of tell when the virus has overcome that resistance and the cells drop a little bit, and then a new resistant mutation arises. But at the end, or even in the middle, it's really complicated by a lot of different genotypes within both the hosts and the viruses. We're keeping it at the same nutrient level, right? Same light level, same diurnal cycle. There's been studies where they've changed, nutrient level, and you see completely different dynamics. I don't know, Debbie, do you want to add anything about coevolution? Yeah.
No, that was fine.
Debbie, I have a question about the reservoir. I'm really interested in the finding that there's all of these viral genotypes that are present in the virome that you don't see present in the metagenome, that they're not infecting. How long do you think they're there? You've done some work on decay rates, and how are they maintained to sort of wait out until they find, a particular host that they want to infect? I'm really interested in this.
Yeah. I mean, I think it's a really hard question for a field study, right? And I also, I don't know that our level of detection at meta-G and viromes is at a level we can say that there's no replication of the rare types. And I think that there has to be some level of replication over a period of time, because the decay rates, at least that we do in the lab and in the field, are on the order of days. We were talking about particles, days to weeks when we're talking about particles, much less when we talk about infection. But when we're looking at meta-G and meta-T, and also the Polanyi method for quantification, we're actually looking at particles. So they have to be meeting a host within I would say, a couple of weeks to a couple of months. Otherwise, they're going to be lost entirely as particles as well. But it's clear that there's, at least within the T4 likes, and we think about it in terms of T4 likes have both specialist phages and generalist phages, that perhaps there's a lot of generalist phages hanging about. and that a particular type can infect a particular cell that comes along. And if that cell is dominant, then it can replicate rapidly and become a very dominant type, even though it's quite minor in the environment until it bursts. And it depends on, you know, whether because we didn't do a time series in that was a transect. So we've moved on. Perhaps we would have seen higher abundance in the reservoir a couple of days later.
Hi, can I ask a question? I have a question for Debbie. So you showed that calculation about carbon and in the hotspot that it could kind of supply a certain percent to heterotrophic bacteria. Did you look at nitrogen or phosphorus, or whether or not kind of that degree of lysis how that could supply or cause regeneration of nitrogen and phosphorus in that zone?
Yeah, we could do a calculation. I don't remember whether we did a specific calculation for nitrogen or phosphorus and just decided to show the carbon or not, but it's basically an estimate, assuming that everything within a certain period of time is going to become biodegradable and is bioavailable, Kim, which we certainly don't know that of. But no, we didn't. I mean, I don't know off the top of my head how it would look if we were to do that by NLP, but we could do that. Yeah. But measuring those types of things is the biggest trick, especially when you've got all this diversity, like what you talked about, Kim. I think that's very cool. It's fun as work. It's very cool that you've got two different viruses and you've got a very, very different lability of the DOM. I think that's complicates all of the modelers' work, but it's very interesting. Do you? I can ask either Kim or Hannah. Do you have any idea of why the viral infection with an RNA virus, the organic matter is not labor?
I'll let Hannah.
You can take it, Kim.
I think it's a good question. We don't know. I mean, it's maybe there's additional processing that happens during like a DNA viral infection that allows the molecules to be smaller and more readily accessible. Like heterotrophic bacteria can only take up, you know, certain size spectra of DOM. And so, you know, the idea is that maybe the DOM is just larger, less processed in the RNA during RNA viral infection. So when it gets out, it's not readily available. I think eventually it becomes available, you know, but it just requires that extra step of processing, which I think does add a layer of complexity, because now you're looking at, you know, not directly supporting secondary production and that time dependence. And so, you know, it all comes down. That's the other thing that we don't really talk about is the time component, right? The time it takes for things to get recycled versus exported. And what is that time? And that's kind of something we stayed a little bit away from, because I think that's going to be a difficult thing to assess. But, you know, how fast things are being turned over is going to really impact how fast it can be exported. So it might not, you know, maybe the extra step of processing doesn't really matter in terms of shunting or recycling material, but it's something that I think we need to, you know, sort of understand, at least from basic level, you know, whether that is going to be able to make it back, you know, make it back into the microbial loop eventually.
You could imagine it might have implications for which components of the microbial community will do better.
Yep.
Yeah. Are the RNA virus infections notably faster?
They're actually slower.
They're slower. Okay.
Yeah. But not
So
by much. Yeah. Sorry.
Do you know the diversity of like how many different types of single-stranded DNA viruses are there? How many different types of RNA? You showed sort of two model systems. Is there a wide range of single-stranded RNA viruses that have different genotypes that might do something different, or?
I think among the RNA viruses, there is a great deal of diversity. We know very little about the single-stranded DNA viruses. Jessica would be able to speak towards that. They're methodologically difficult to get at. You have to process, you know, you have to sequence it in a way that most of us don't sequence. And so I think we know very little about single-stranded DNA viruses. And so most of the work that we focused on has been on the RNA viruses because we can get at those more with metatranscript, like metaomics. But so the RNA viral diversity is very high. I imagine the DNA, the single-stranded DNA diversity is super high as well. I don't know, Jessica, if you have comments about that, but I imagine they're so small and I just feel like, yeah, they could be super diverse.
Yeah, that's the thing with single-stranded DNA viruses. They're very diverse and their genomes are quite small. And most virome studies, they now have a cutoff of 10 kb and the genomes are below 5 kb. So even if we lower the cutoff, we miss out on a lot of single-stranded DNA viruses, unfortunately.
What did you mean the cutoff is when they do analysis, you
Like
mean?
when we analyze viral metagenomes, like when we do total DNA, most people will, when these, like to keep the good quality viral genomes, they have a cutoff of 10 kb, which will
Of
naturally
contigs, but
remove
you could really
all-
look at reads though, right?
You can still look at reads and you can still look at the smaller ones, which we do in my lab. We look at the small ones because otherwise you miss out on all these shorter viral genomes like single-stranded DNA viruses.
Do you need to do like a rolling circle amplification to get at them though? Or are they?
Not anymore. Because we do get a lot of single-stranded DNA viruses in our viromes without doing the whole genome amplification. And I think at the time, like that age yourself without aging yourself. When I did my first viral metagenomes, it required double-stranded DNA to send for sequencing. And that's why we were using the whole genome amplification, which was biasing for single-stranded DNA viruses. But we do just total DNA now, and we get quite a bit actually. If we keep small contigs.
You do any processing before the DNA extraction? I mean, any host removal? And after sequencing also, do you do any, how do you remove the host reads? Before the sequencing and after, do you do anything to, get better virus?
I can answer this, but because that's what we do always in the lab, but we always pre-filter to remove potential hosts through a 0.22. And then before doing, then we concentrate the viruses, and before doing the extraction, we usually do a DNase treatment to remove free-floating DNA. Then we do the viral extraction. And then once we get the sequences, we use bioinformatics to keep only the viral contigs, even if we should technically have only viral contigs.
Yeah.
Anyone have a last burning question? No? Well, oh,
Sorry,
David.
keep everyone on the
Yes.
line. I was just interested with Kim's talk, those changes in the SI to C ratio that you find. Maybe you showed this and I missed it, but there are also changes in particle size, right? Because you think for export that that's the size of the particle would also matter. Just curious if you could comment on that.
Yeah, I mean, I think we see, I think the increase in particle size comes post lysis. And so when you have, sort of all that sticky intracellular contents that come out, that causes the aggregation. The increase in elemental like stoichiometry we see in intact cells. And so they're not yet. So that's pre-lysis. And that's likely due to just a slow growth rate. So diatoms, whenever they slow their growth rate, if there's enough silicate in the environment, they'll just keep on taking it up and producing their silica wall. So I think you're talking about two different times within the infection cycle.
Would you imagine the aggregates being more ballasted in a nutrient limited, if the cells
Yeah, I think it comes down to the time, right? And so you get this increase in SI to C in that early infection, and so how fast that material lyses then and then is able to aggregate, and then that ballast would definitely cause the material to sink faster.
I would like to thank our speakers. Those were great talks. And also, this concludes our second webinar. And again, if you guys are interested in joining, even if it's remotely, the in-person seminar, the workshop in June, you can sign up for it. And if you have any other questions, well, you guys know where to find us.
Thank you.
Thank you.
Yeah.
Thank you.